Hi everyone, I'm Claudio Argentino. I'm a researcher at the Arctic University of Norway. In this video, I'm going to provide you with a brief introduction on coal seeps and on the biogeochemistry of methane in marine sediment. By definition, a coal seep is an area of the seafloor characterized by the emission of hydrocarbon-rich fluids. Hydrocarbon that can be gas, like in this video, mostly methane, or oil. When we look at the global distribution of coal seeps, we soon realize that they mostly occur along continental margin. You see from this map. This map is based on water column observation of train of bubble rising toward the sea surface, which generate these acoustic anomalies known as gas flares. And this map also includes direct seafloor observation of bubbling. You may also notice that a few areas in the world appear as hotspots of coal seep. But these likely are biased by the large number of studies conducted in this area for coal seep research. Coal seep distribution tightly matches with the sediment distribution. At continental margin we find thick piles of sediment rich in organic matter. And this organic matter can undergo maturation, so there's higher potential for hydrocarbon generation. And consequently, some of these hydrocarbon can leak from the reservoir and migrate to the seafloor. This other map is showing a different aspect of this organic matter cycle. Specifically the flux of TOC, total organic carbon, toward the seafloor. So it's the flux of organic matter actually reaching the seafloor and being buried. And we see, again, that the organic carbon flux is higher along continental margin. We said that the hydrocarbon eventually can leak from the seafloor. So how they reach the seafloor? Well, you know, hydrocarbons are buoyant, so they tend to rise through the sedimentary column. And they follow mainly two types of pathways. A structural pathway can be a thrust, a fault system, fractures. But they can also follow stratigraphic pathways. Like in this image, we can see a stratigraphic pathway can be a high permeability layer, like a sandy layer enclosed in muddy formation, which are low permeability material. And they also follow the geometry, right? Because the hydrocarbon migrate in the sandy layer toward higher parts of it. And they can eventually be trapped again against impermeable barriers, or they can find their way up to the seafloor. But gas can also be trapped in the sediment in the form of gas hydrate. Gas hydrate is a unique type of material, looks like ice, and it's indeed composed of water molecules forming a cage, a crystalline lattice, and this cage is hosting methane in it. So actually it's stable only if it has some methane in it. And as you can see from this video, it can catch fire very easily because of its high methane content. And this material is stable within the sediment under specific conditions, generally very high pressure, so a thick water column, and low temperature. If we look at this graph here, so we have depth and we have temperature. So when we move down into the water column, the temperature drops, right? And the pressure increases. And then we encounter the seafloor. the temperature tend to rise again due to the geothermal gradients, right? So increase, increase, increase. And there's another line here, which is the phase boundary for methane. So we find methane dissolved in the pore water and available for gas hydrate on the left. And we see that gas hydrate stability in the sediment is confined to a very narrow interval marked by these two stars. So where the seafloor, from the seafloor to the intersection between the phase boundary and the geothermal gradient. So this is the interval in the sediment where we expect gas hydrate to be stable. And again, the stability of gas hydrate is higher at continental margin, where we have a lot of hydrocarbon in the sediment, so high hydrocarbon concentration. We have generally high pressure and low temperature. We don't find the gas hydrate, or we don't expect gas hydrate to be stable in the middle of the ocean. You see, no gas hydrate stability. Even if we have high pressure and low temperature, right? The reason is because there's not enough gas generation. There's not enough gas in the sediment to stabilize the gas hydrate structure. Should we be concerned about the flux of methane reaching the atmosphere? You know, methane is a strong greenhouse gas, and if it reaches the atmosphere, it can contribute to global warming. Actually, the contribution from coal seeps is minor. You see, from 3 to 6% compared to total natural emission. And the reason is that the methane bubble rising through the water column get redissolved and oxidized in the water column. So actually, only in a very shallow coastal environment where you have a short water column, we have the higher contribution from coal seeps, because the water column filter is reduced. But there's another very important filter for methane in the sediment. It is actually a biofilter because it's performed by microbial consortia, including archaea and bacteria. You see, they work together in a cluster. And these microbial cluster, they are mostly concentrated in a narrow interval of the sediment called sulfate methane transition zone, because they actually utilize sulfate to oxidize the methane anaerobically. And the product of this reaction are bicarbonate and hydrogen bisulfide. So actually, this sulfate methane transition zone is releasing hydrogen sulfide, which migrate toward the seafloor and sustain very peculiar chemosynthetic communities. So we said these areas are generally deep enough to be dark, sometimes completely dark. So there's no possibility to perform photosynthesis down here. That's why this organism got specialized to utilize chemical energy instead of solar energy to perform their metabolic processes. So basically, microbial meth or higher organism can reoxidize the hydrogen sulfide back to sulfate, and that what release some energy that they can use for their metabolism. And actually, the depth of the sulfate methane transition and the flux of hydrogen sulfide to the seafloor are controlled by methane fluxes from below. So the higher the methane flux, the shallower is the sulfate methane transition, and the higher is the flux of sulfide. And where this flux of sulfide is very high, we find generally microbial meth, white microbial meth, because they can tolerate this stressful environment. Whereas where the flux is lower, hydrogen sulfide flux is also lower to the seafloor, we find higher organism. We have an example in this video. We were trying to collect a push core with the ROV from a microbial mat. So this is the microbial mat you see, and it's surrounded by bushes of tube worm. You see there's a clear habitat zonation, we can say, and called seeps. So we might expect higher methane fluxes and shallower sulfate methane transition where we see the microbial mat. I wanted to talk a little bit about the origin of methane, right? We said hydrocarbon potential is high along continental margin, so organic matter can produce hydrocarbon. But actually there are other type of methane sources. So if we look at this image here, we can simply classify the type of methane into two categories, biotic or abiotic. So biotic means it comes from organic matter. Abiotic means it doesn't come from organic matter, but from other inorganic reaction. If we look at the biotic methane, it can be further classified into microbial or thermogenic. Microbial means the organic matter is being degraded by microorganisms to produce methane. Thermogenic means the organic matter underwent some thermal cracking, some physical cracking of the organic molecules without intervention of microbes. You also see that I mentioned biogenic here because it's still a common term used in the literature, but it can be ambiguous in certain scientific fields, earth science fields. So we prefer to stick to microbial term. What about abiotic? Well, abiotic processes generally are related to volcanic areas due to degassing processes, one source, but also to other deep processes related to serpentinization reactions, so basically gas, water, rock interaction. And we are not going further into these abiotic sources. We mostly focus on this biotic part for the rest of the video because it's the most abundant gas in the field worldwide. Regarding microbial gas, it can be produced by two different pathways: carbonate reduction or acetate fermentation. They both occur pretty shallow in the sedimentary column, you see, within a kilometer or less. This is mostly microbial gas. Whereas when we move deeper, we generally encounter thermogenic dominated gas from cracking of organic matter. And we see from this plot here. And it can be, as you see, also associated with oil. So in the same reservoir you can find oil with some thermogenic gas. In this pie chart here, we see in the gas field worldwide, generally the thermogenic gas is the dominant, whereas the microbial is minor. Actually, we find another new term here, secondary microbial gas, which refer to gas derived microbially from petroleum, from oil biodegradation. And it's still a minor component of the gas field worldwide. All right, so how can we distinguish if gas seeping out of a sediment is thermogenic or microbial or abiotic, for instance? So we have to rely on geochemistry this is one of the tools we can use. We can collect gas and then measure its isotopic composition, for instance. Basically, we are looking at isotopes. The concentration of the heavier isotope of carbon and hydrogen, so deuterium, compared to the concentration of the lighter isotope, carbon and hydrogen. So we are picking carbon and hydrogen isotopes because the methane molecule is composed of carbon and hydrogen atoms, right? So we obtain a parameter which is a delta C13, and this parameter is plotted here for methane, and the delta deuterium is plotted here for methane. So in this plot we see the different gas field, origin field, so microbial in blue, and then we have abiotic, we have thermogenic, and also the secondary microbial. So you see, we can start distinguishing based on this isotopic plot. So what's the reason why we find microbial gas pretty depleted in the heavy isotopes? Very negative isotopic value. For a reason which is called kinetic fractionation, so microbes prefer to break molecular bonds where the molecules are composed of the light isotopes, because these bonds are weaker than the bond among heavy isotopes, right? So they prefer, it's more efficient energetically to break this bond between light isotopes. So as a consequence of this, the methane released is going to be pretty depleted, mainly composed of very light carbon and also hydrogen isotopes generally. Whereas the thermogenic process is less specific, right? Because temperature is breaking molecules with light and heavy isotopes. Yes, the light isotopes bond are weaker, so there's still some preferential breakage of this bond at higher velocity compared to the heaviest one, but it's less specific as for microbes. So the thermogenic field, you see, generally has heavier delta C13 in the methane and also heavier delta deuterium, right? So the first tool, isotope geochemistry. But the second tool is the molecular composition of the gas itself, because gas seeps are, as I said, dominated by methane, but they generally include also higher hydrocarbons, ethane, propane, other hydrocarbon gases. If we look at the relative abundance between methane and the heavier hydrocarbons that we can plot as a ratio here against the delta C13 of methane, we also see another way to discriminate between the gases. The microbial gas tend to be drier, so dominated by methane, because they release mostly methane from their processes. Whereas the thermogenic and the other origin field, you see, also cover the very wet gas. Wet is a term that means the gas includes also a lot of heavier than methane hydrocarbons. So microbial gas tend to be dry, methane dominated. Regarding the biodegradation of oil and methane from secondary microbial processes, there is one way to find out if there's some biodegradation going on. You see that from previous plots also the isotopic and molecular composition sometimes overlap, especially for these microbial gas of secondary origin. But if we look at the delta C13 of the carbon dioxide in the seeping gas, we can easily find out if there's some gas from biodegradation of oil, because it has CO2, which is pretty heavy isotopically. So this is a third plot we can use to figure out the origin of our gas. So now I end up with this slide showing and introducing the exercise. So I mentioned that we collect gas escaping the sediment. You see, we use this bubble catcher here. The gas is conveyed into a funnel and then it goes into bottles. And we measure the isotopic and molecular composition of the gas. So I give you a couple of examples, a couple of areas. Let's say area A, area B, and 2 gas samples from each area. We have the delta C13 of methane, we have the delta deuterium of methane, we have the molecular ratio of the gas, and we have the delta C13 of the carbon dioxide. So based on this geochemical data set, I would like you to start reasoning about the possible origin of these samples of the gas from these 2 areas. Is it mostly microbial? Is it thermogenic? So you start just by reasoning behind this, based on previous information I gave you. And then you will try to plot these samples on the tree plot with the origin field. And then we will discuss the interpretation in the next video.